So we're moving on to our last topic of the year, which is genetics and evolution, and this will also carry into your Year 11 science next year, regardless of which topic you're taking, with chemistry, with bio, with physics, doesn't matter, you will have to do genetics again next year, and it really builds on this. So it's really important that you guys start to get this year, get the basics down, and then next year you can build on that to work towards getting those merit and excellence credits. Okay, so you've already done the brainstorm about what you already know about genetics, so make sure you take a second to go back and read what everyone else has brainstormed on there as well. Okay, so learning intention 1 is what we're going to work on today, and this is about the structure, the function, and the location of DNA. So what is DNA, where do we find it, and why is it so important? So we need to look at DNA, what's the definition for DNA, what's the complementary base pairing rule, where do I find it, and what is the shape of DNA. Then we'll have a look at what it's made up of, and these special things called nucleotides. Okay, so what is DNA? DNA is the shortened word for deoxyribonucleic acid, which is a huge word. You don't necessarily need to perfect your spelling of this, but the word itself actually helps us with figuring out what DNA is made up of. So we've got the deoxyribo is the sugar, and then we've got nucleic acid, which is the bases part of it here. Don't stress too much about learning this name. DNA, capital DNA, is fine for us at this stage. Okay, so one of our points for achieved was that we know where DNA is found. So DNA is found in the nucleus of a cell. So if you think back to your botany topic last year, if we zoom in on one single cell, we've got that main part of the cell called the nucleus, and we've got all these other different organelles sort of floating around the cell, but the nucleus is the really important part of the cell that gives all that information for the cell to keep doing whatever its job is, like a skin cell or a brain cell or a liver cell. Whatever it is, it needs the information to know how to do its job. So DNA is that information, so it is the genetic information. So we find DNA, this genetic information, in the nucleus of the cell. And this information that's encoded for in the DNA is used to control the structure and the function of living cells. So like I said, to tell the cell what to do. So it's the instruction manual to make new cells. So the way that you can sort of think about this is if you ever bought like IKEA furniture or flat pack furniture, you get all the different bits, but how do you know how to put it together? So the cell's got all the different bits already there in the cell, but it needs some information or some instructions to tell it, how do I put these bits together so that it functions as it should? So you might get all the bits to make a table, but you need the instruction manual to tell you how to put those bits together so that your table functions as a table and doesn't fall apart. So DNA is that instruction manual. It tells the cell what to do and how to do it. Okay, our next bit of keywords is a chromosome. So what a chromosome is it's just a really long piece of DNA that's wound up tightly. So here's my piece of DNA that's really, really long. If I just wind it up all tightly like this, that's a chromosome, a really long piece of DNA wound up tightly. So I've got just like a piece of string that you can wind it up really tightly to make a thick rope. That would be like the chromosome. And the reason that we twist our DNA up into such tight little structures called chromosomes is because it would really just get knotted and tangled with each other if we just left these long strands of DNA just floating around the nucleus. So it winds up nice and tightly to keep it organized and keep it from tangling up with other pieces of DNA. So keywords so far: DNA is the genetic information, the instruction manual, Chromosomes are long wound up pieces of DNA. Okay, so the next part of our learning intention is, what does DNA actually look like? What's the shape of DNA? So we call DNA a double helix structure, and when you do the next activity that's set for you, you'll start to get a better idea about this. So when I say double helix, what we've got here is two connected strands of DNA that wrap around each other. So they look like this, the two strands, but then they twist like this. So you can see this double helix is these two strands that sort of twist around each other like that. So you can see it a bit better here. Okay, so you can really see that shape in there, the double helix. Okay, so DNA, we're building on this definition now. It's the genetic information of our cells, and it's in the shape of a double helix, which is those two strands sort of wound around each other. And what DNA is actually made up of is separate building blocks, and we call those building blocks nucleotides. So in a nucleotide, we've got three things. We've got a sugar, a phosphate, and a base. These are the three things that make up one nucleotide, and then thousands of nucleotides back to back to back makes up our strand of DNA. Okay, so we can see here we've got the phosphate, the sugar, and the base. So this is one nucleotide. Here is another nucleotide. Here's another nucleotide. Oops, sorry. Include that phosphate. And another nucleotide. So you can see one phosphate, one sugar, one base makes up a nucleotide, and these repeat along the strand to make up a DNA strand. And what we've got here is the backbone of the DNA, that sort of outside bit, is the sugars and the phosphates. So just like I've got here, this is kind of like having the hard cover of the book. The point of having the hard cover is to protect the pages inside, which actually has the information on them. So the bases here that go down the middle, this is where the actual information is stored. It's those bases in the order of these letters that we'll look at soon that encodes the information to tell your body how to do its job. The only reason we've got the sugar and the phosphates on the outside is to protect that inside layer there. So you do need to know that a nucleotide is a sugar, a phosphate, and a base, and you would need to be able to circle one nucleotide on a diagram. Okay, so you can sort of see in the middle here, so we said a nucleotide is a sugar, a phosphate, and a base. When I say base, there's 4 different types of bases here. You don't need to memorize these names like adenine, thymine, guanine, cytosine. You don't need to know those names, just these letters would be good. So we've got A, T, G, or C. So we've got 4 options for the bases here. So we've got a sugar, a phosphate, and one of these bases and one nucleotide. Now, one of our learning intentions was to identify the complementary base pairing rule. And the rule here is that A always pairs with T and C always pairs with G. So there's a few ways to help you remember this. GCAT, so G and C, A and T. If you're more of a visual person, the curvy letters, the C and the G, go together, and the sharp letters, the A and the T, they also go together. So if we look at the diagram, we've got a T and then an A. A, then we must have T. C, then we must have G. So they always pair up following this rule. And we'll look at this later, but the reason you've got this rule is because this information is so important, we really can't risk losing this information, having a mistake. So if your DNA unwinds and you've got this one strand left on its own, you don't want to have just a single copy, and if something goes wrong, well, that's it, you only had one copy. Okay, it's safer to have 2 copies so that if something happens to one of them, it's okay, we've still got the other strand. So we've got 1 copy here and another copy here. Following this base pairing rule, we would be able to fill in the blanks if we lost one of these strands.